Hello, my name is Sofia Notopoulou, and this is my poster presentation regarding a multi-omics analysis in a neural stem cell model of Parkinson's disease that provided us valuable insights into the disease mechanisms. A few words about Parkinson's. It is the second most common neurodegenerative disease and the fastest growing neurological disorder for people over 65 years old. Its main symptoms are tremor, rigidity, and bradykinesia, as well as some non-motor symptoms. Regarding histopathology, there is a selective degeneration of dopaminergic neurons located in the midbrain. During disease progression, these neurons are full of insoluble aggregates known as Lewy bodies, which are mainly consisting of the protein alpha-synuclein. Mutations in the responsible gene, SNCA, account for the earlier onset of robust symptoms. Nevertheless, the pathological mechanism of alpha-synuclein remains elusive, hindering the development of effective treatments. So, our aim was the generation of a reliable cellular model of aggressive Parkinson's disease for in vitro and in silico modeling of the pathological mechanism of alpha-synuclein. We modified neural progenitor cells to reducibly overexpress the mutant SNCA-53T for 2, 5, and 10 days, corresponding to early, middle, and late-stage pathology. Cells cultured in the absence of dogs were the control sample. Day 5 was the best time point to study, as we noticed events but not irreversible pathological events. So we designed common in vitro experiments for the characterization of the pathological responses in day 5 versus day 0, and in parallel, we ran proteomics, metabolomics, and lipidomics analysis for the identification of all differentially produced proteins, metabolites, and lipids in these time points. By combining all these outcomes, our final goal was to depict a representative snapshot of the dysregulated systems biology that emerges during Parkinson's disease, which will be valuable to better understand disease pathology. Cells cultured with dogs for 5 days produced high levels of alpha-synuclein A53T, while cells cultured without dogs were negative for the protein. Interestingly, observation of cells in high magnifications revealed multiple abnormal vacuoles in the cytoplasm, indicating a pathological cellular condition. NPCs were further differentiated into neuronal populations, exhibiting the characteristic neuronal morphology. However, those cultured with dogs and overexpressing the SNCA A53T for 5 days had lower differentiation potential, with their axons being thinner and more fragile than control ones. In vitro characterization saw a significant reduction of energy production on day 5 of alpha-synuclein A53T overexpression, which was accompanied by acceleration of reactive oxygen species production. Additionally, there was elevated programmed cell death by apoptosis and ferroptosis. Proteomics analysis identified 359 significantly dysregulated proteins in alpha-synuclein A53T overexpressing cells compared with control ones. The pattern of dysregulation effectively discriminated the pathological from healthy cells, and in general, the dysregulated proteins can be clustered in 4 major groups for unfolded protein response, expected mechanism due to alpha-synuclein aggregation, estrogen signaling, a group of hormones that induces alpha-synuclein aggregation, Parkinson's disease, validating the reliability of our model, and fatty acid metabolism. Analyzing the dysregulated proteins in more detail, we retrieved the exact key pathways that were affected due to alpha-synuclein A53T overexpression, with cholesterol metabolism being among the top hits. Looking closer into the pathway, we mapped the exact dysregulated proteins, and we noticed that some of them are actually biomarkers for risk of development Parkinson's disease. Due to the remarkable impairment of metabolism in our Parkinson's disease model, we ran a metabolomics and lipidomics analysis for the identification of all dysregulated metabolites and lipids. The affected molecules participated in the metabolism of nicotinate and nicotinamide, derivatives of NADH coenzyme, the recycling of several phospholipids, and the biosynthesis of amino acids. Interestingly, all the dysregulated metabolites are representative for brain tissue. Integration of proteomics, metabolomics, and lipidomics data through a regulatory network revealed that there are 259 proteins, metabolites, and lipids, which are highly associated and promote Parkinson's disease. Molecules are clustered into 8 groups, each one associated with a particular biological pathway. The network indicated that fatty acid and glycosaminoglycans metabolism, ferroptosis, and nicotinate and nicotinamide biosynthesis are highly connected with Parkinson's disease. Also, there are some central proteins that play an important role in the information flow with Parkinson's disease molecular network. So, neural progenitor cells are a very good option for study neurodegenerative diseases, and especially SMNPCs overexpressing the SNCA-A53T mutant make a reliable model for Parkinson's disease, as they recapitulate the characteristic pathological responses in vitro. Through proteomics analysis, we noticed a dysregulation of lipid metabolism, which was also validated by metabolomics analysis. Specifically, there is a significant impairment in the mitochondrial and membrane-associated lipids. Integration of follow-omics data and network analysis highlights that Parkinson's disease is interconnected with nicotinate and nicotinamide metabolism, glycosaminoglycans degradation, fatty acid metabolism, and ferroptosis. Thank you for your interest, and I'll be happy to hear from you.